Good morning, all. Today we'll start with the first session. We have with us Professor Deepshika Chakravarti. She is a professor at the Department of Microbiology and Cell Biology, Indian Institute of Science, Bangalore, and currently holds an Astra Chair professorship. She is a Humboldt Fellow and did her research in FAU, Erlangen, Germany. Professor Deepshika did her master's in microbiology from Nagpur University and a PhD from National Center for Cell Science, Pune. Her research focuses on infectious disease and vaccine development. Her work has been published in top biological journals, including Journal of Experimental Medicine, EMBO Journal, Journal of Immunology, Infections and Immunity, Journal of Clinical Microbiology, AAC, Journal of Antimicrobial Chemotherapy, PLOS Pathogens, etc. She has won many awards to her credit, like Tata Innovation Fellowship, Astra Chair Professorship, SK Chatterjee Award, DAE Outstanding Investigator Award, National Bioscience Award, DBT, NASI Reliance Partnership Platinum Jubilee Award, etc. She is a Fellow of National Academy of Science, India, Indian National Science Academy, and Indian Academy of Sciences. She is involved in editorial board of many journals like Virulence, Innate Immunity, PLOS 1. She serves as a reviewer in reputed journals like Lancet, Nature Communications, MMBR, to name a few. Her research has been supported by grants from DBT, DST, ICMR, DAE, etc. She has more than 150 papers published in international journal and has guided 20 students so far towards their PhD. 10 more are continuing PhD currently. More than 200 summer trainees and 60 academic summer trainees have worked in their lab. Most of our PhD students are faculties in reputed institutes like ISC, BIIS, Pilani, NIT, etc. Professor Deepshika is a passionate teacher, a mentor, and she aims to teach microbiology to the children and general audience and to bring out the importance of microbes in day-to-day activities. So with this short introduction, I invite Professor Deepshika to deliver it. The floor is yours.
Thank you, Dr. Murugan, for your very humble and very nice introduction. It was not short though. Okay, anyway, so this is my second lecture, right, Dr. Murugan?
Yes, Professor.
And I had actually enjoyed my interaction the first time, and so I'm looking forward for more interaction today. So today I am going to talk to you. Let me share my screen. Can you all see my screen?
Yes, Professor.
Yeah, great. So, and you can see my full screen also, right?
Yes, Professor.
Okay. So if anything, you know, that you cannot hear me or you cannot see me, you cannot see my screen, kindly, Dr. Murugan, you can kindly stop me immediately. Yeah.
Okay, Professor.
Thank you. So good morning, colleagues, and I hope that you are very fresh in your mind, had your nice breakfast, and let us discuss something which is very close to my heart, and it indeed talks about the stealthy approach of a pathogen of infectious disease to completely subvert the host immune system. So the topic that I'm going to talk to you talks a lot about the beautiful ideas that you can also get it in your mind when you're working with infectious disease and immune system. And the question is that in spite of having important immune system, why do we become sick? So a colleague of mine, Mayuri, who was a PhD student with me, she was an integrated PhD student. She did this wonderful work, which I'm going to present today. And currently she is a postdoctoral fellow in the MRC Cambridge, UK, and extremely doing well there, again, working in the field of immunology. She is the one who, in fact, started working with one of the most difficult but most important cell type, that is the dendritic cells. The dendritic cells are the cells which are efficient antigen presenters. Unlike macrophages, which may present but which may also degrade, the dendritic cells are specialized to present your antigen in a very robust manner. And if we do not have a good proportion of the dendritic cells, then the immune system will become totally compromised. But here, let us see that how this Salmonella behaves when it sees the dendritic cells. 1 minute. Slides on. Okay. So why this disease Salmonella? For that matter, you can be working with any disease, and the same rule will apply, or with some kind of deviation will apply. So just take it as a model system. Now, why Salmonella is so important is because of its widespread nature. If you look at the prevalence, look at that complete red, the India, Asian countries, where the enteric fever burden is extremely high. And if you look into some of these countries like, Australia or USA, there the burden is quite low. Look at the South America. Here also the burden is extremely high. Now it is coming very high, though it is with a medium burden, but you will not see the countries, most of the countries where there is no burden. Of course, here you see all white. That means there is no burden, but the sporadic cases of Salmonella, because of the increase in the travel, is increasing. And that is why the CDC has put this Salmonella as a bioterror category number 3. And it's a global agenda to make a vaccine against this. So it's a very good model system. How does this bacteria makes us sick? Now, from our previous talk, this is a reiteration so that you all get refreshed with your memory of how Salmonella make us sick. Now, unlike an aerosol pathogen, like Mycobacterium tuberculosis or say Klebsiella or Pseudomonas, Salmonella can only make oneself sick if you take it through the contaminated food and water. So if we ingest a contaminated food and water, then it gets inside the gastrointestinal tract, and it passes through the acidic pH of the stomach, a very low pH, around 2.3 to 2.5. It sustains there very well, and then it reaches a small intestine. Okay. Now, small intestine is a very interesting site and a hub for many of the gastrointestinal pathogens, bacteria, viruses to be taken in and cause the disease. Now, what is it in the small intestine, and why the bacteria or the viruses should take this niche? Because, small intestine life is not so simple. The intestinal layer. Now, what is small intestine made up of? It is made up of these enterocytes or the columnar epithelial cells. And these cells are very, close to each other, and they're adhered by something called as a tight junctions. And that is what gives it an integrity. Now, the intestinal epithelial cells are protected by a mucus layer. So there is, let me put it into a point scale mode, pen mode. So now this epithelial cells, actually, they are protected by a mucus layer, somewhere around 100 to 120 micrometer thick mucus layer. And on top of this mucus layer, you see the gut flora, the very famous second organ, what we call about our microbiome, that is the gut flora. So there are more than around 230,000 different species. It is composed of a wonderful diversity of bacteria. Of course, there are certain phages, bacteriophages, certain yeast, fungi, and they all make the correct flora. Now, the mucus separates your gut flora and the cells. This you need to keep it in mind. Now, how the pathogen can compete with such a robust flora and still get inside? Now, here comes the trick which is employed by the pathogen, like Salmonella. Among the enterocyte lining, you have certain cells which are known as a M cell. Now, these M cells are named because they are the membranous cells or the microfold cells. These M cells, they throw their kind of arm up, and the bacteria slips in through these M cells, because this particular area is an area of no mucus. So if there is no thick viscous mucus present, the bacteria need not compete with anybody. It just slips in. And once it slips in, it undergoes a transcytosis from the apical side to the basal side. Yeah, so this is the apical side, which is facing the lumen. So consider it as a rod. All the cells are from the apical to the basal. Now this lumen is the one where the bacteria has entered. So it will have to undergo now transcytosis from the outside to the inside. And during this process of transcytosis, the major cell which helps in the bacteria are macrophages, dendritic cells, the T cells and the B cells. Now all these cells should have been killed the bacteria as its nature is. The macrophages, the moment it sees a pathogen or any particulate matter for that matter, it is going to, its job is to destroy. But why you call a bacteria as a pathogen? When does it start becoming pathogen? The day or from the time it is somehow in the arms race or a tug of war to survive itself, but the cell will not allow it to survive. It is its destiny is or destination is to kill whatever is coming, but they want to survive. From there, when the tug of war begins, the arms race begins. And that is where all the evasion strategies begin. So now the macrophages, instead of killing them, what they do, they use this, the bacteria use them as a vehicle to transmit from the intestine now to all the distal organs like liver, spleen, mesenteric lymph node, to the kidneys, etc., etc. So these are extra-intestinal infections. So that is how the Salmonella pathogenesis is caused. Now, if you now keep it in your mind that the dendritic cells or the macrophages, specifically the dendritic cells, what is the job of the dendritic cells? Their job is to present the antigen. Whatever comes to them, they will not immediately destroy it. So this brings to the important arm of immunology and the most important immune function in any system, which gives rise to protection. And that is a basis of your vaccination to work. That means the antigen presentation pathway should be so robust that once the antigen is presented and processed, you will have a memory cells, and then whenever a challenge comes, you get protected. So that is what is antigen presentation. Now, all of you should look back to your maybe graduation or post-graduation teaching in immunology and just recall the antigen presentation. How is the antigen presented? First of all, what is an antigen? The antigen is a molecule which is foreign to the body. That is an antigen. That means the host has never seen it. So that is known as an antigen. It is not necessary that every antigen will have a antibody response or a cell-mediated response. But yes, majority of them have. What is the class?
Excuse me, ma'am.
Yes. Yeah.
Ma'am, can you, ma'am, I am just starting to understand this part of the biotechnology area. So
Okay.
can you just go a little bit slower, or maybe start with the basic of it so that I understand it, because I am sitting in my department.
Okay. No, but tell me, first of all, you should tell me what is your background?
Background, I am just a beginner, ma'am.
No. What is your subject?
Biotechnology department.
Biotechnology. You are in your first year.
No, third year.
So in third year, this is strange. The antigen presentation should have been taught to you in first year. How is it
Antigen
possible?
virus virology. Ma'am,
No.
classes were online. Classes were online. We were not able to understand what is being taught because we had at our home Wi-Fi issues or something like that. So
Okay,
what can
tell
we
me,
do?
what is your name? What is your name?
Tushar Verma, NIT Durgapur.
Tushar. Okay, you're from NIT. I used to get students. Tushar, I need to take extra class for you. Let me tell you.
Okay.
Just to tell you what is antigen presentation, by the dendritic cells. First, you need to know what is a dendritic cells. The dendritic cell class itself, that from where the dendritic cells originated, who discovered dendritic cells? The dendritic cells were discovered by none other than, do anybody know in the crowd who is the person behind the discovery of the dendritic cells? A very famous person who kind of got his award posthumously, his Nobel Prize. Anybody?
Ma'am, Ralph Steelman.
Very good. Ralph Stillman. Very good answer. Now, Ralph Steinman was the person who was a medical doctor. So he was in the Rockefeller, and he realized that there is certain specialized cell type which is not going to, not like macrophages. They are different. They show dendrite-like structure, but very strangely, their percentage, the naive percentage in the cell, or in the, not in the cell, but in any organ, say in the spleen, secondary lymphoid organ like spleen, mesenteric lymph node, etc., they are very low, like 0.1%. Now you can imagine that such a low 0.1% of the cell, what it has to do with the majorly taking care of immune system. That is where Ralph Steinman's first paper, along with his own mentor Cohen, was the first paper was in Journal of Experimental Medicine, which was a very beautiful paper. If you get a chance, please go through that paper. How beautifully they just observe very methodically these kind of cells. And he is the one who named it as a dendritic cells. Okay. Now, dendritic cells is not so simple. You have a dendritic cells which are tissue specific, like you have a dendritic cell on the skin, which are known as a Langerhans cells. You have a dendritic cells in the spleen. You have subtypes of the dendritic cells in every organ. Okay. And how are they differentiated? They are differentiated on the marker. Dendritic cells chapter itself takes me when I teach my essentials in my immunology. It takes me 3 full class in my host pathogen interaction, which is equivalent to 3 hours. Okay. So maybe Dr. Murugan needs to do something about it for all these participants, for them to first understand whether their basics in the immunology is actually clear. As Tushar has very honestly pointed out, which I really appreciate, Tushar, that in first semester, or in even second year, the different type of cells were not clear to you, not made clear. That means that the entire very important arm of immunology is somehow not touched. Now, if that is not touched, there is no point in understanding what is immune system. But still, what I will do is, because I just have 45 to 50 minutes, and now what is the time? Now the time is already 11:20, and I have few minutes to share. I will put a deal with you. I am opening myself for you to take extra class whenever you feel like. Because I have started my own teaching, I will only have classes I will only have time in the evening, or maybe a late night dinner. So you can approach me and I will teach you in detail. Okay, Tushar, got it? Deal?
Yes, ma'am. Yes, ma'am.
Yeah. Just approach and I teach you the details of host pathogen interaction, immunology, etc., etc. Okay.
Ma'am, excuse me.
Yeah, hello. Yes.
Ma'am, sorry to disturb you.
Yes, Amisha, tell me.
Ma'am, after the session, ma'am, can you please tell me some opportunities and good opportunities that we go for after completing my MSc Microbiology? Like, ma'am, some internship programs and training.
Yeah. I'm going to do that. I will do that.
Yes,
so what I'll do is I'll talk about the science a bit more now, and then I will open up. So what we will do is we will have it question and answer session, so all of you understand. But let me just give you that glimpse of science, so that you are actually interested to know that why the pathogens make us ill. Yeah. So then we open it up. I will keep ample time for the question and answer session. And for all of you who really want to have much more knowledge, kindly approach me. Maybe sometime I will open up and then we can have a Teams online, but I will make it a point for you to understand it, really understand, because it is very important. Now, if you understand this very classical way of antigen presentation, which actually is taught in BSc final year, if you're doing your graduation, post-graduation, or in MSc first year, or for biotechnology students, it is given, the glimpse is given already in the first year. So what you have a very classical MHC class 2 pathway, classical MHC class 1 pathway, Now, I hope that you know the difference between a class 1 and the class 2. If you do not, let me give you in one sentence that the MHC class 1 and the class 2 major histocompatibility complexes, these are the very important molecule which will carry your antigen and present it onto the dendritic cells. Now, what is the difference between the class 1 and the class 2? In the class 1, you know, it is like a structure. It is like two arms. Okay, in class 1, only one of the end of your so-called the carboxy terminal is integrated onto the membrane. And the rest is forming a dumbbell shape and it is closed. So the groove is closed. That is what it is, class 1. So because the groove is closed, your peptide length cannot be very large. It is around 8 to 10. So because it should sit in the groove. Okay. Now, in case of MHC class 2, the groove is open. It's like open like this. So the peptide can be very long, around 20 amino acid to be presented. Now, what is, and it's a big process of this antigen presentation when your antigen is taken up. As I told you, antigen is foreign, right? So you have, say, pathogen, which is taken up. You can have exogenous floating antigens, which is taken up. Food antigens, many of them, you know. Why some of you are allergic to the food, getting hypersensitivity reaction, because these are foreign to the body. Body has not seen it. It will be taken up. So anything which is taken up by these, say, pathogen is taken up by the help of phagocytosis, or your exogenous antigen is taken up with the help of endocytosis. These are the clathrin-coated pits. These are a mechanism, one of the mechanisms by which the endocytosis is happening. That means taking up of any of the foreign particles. Once they are taken up, they land up in something known as endocytic vesicles. Okay. Now, in these endocytic vesicles, from these endocytic vesicles, there are processes which will allow the pathogen to be degraded, right? Not completely degraded that it will undergo nothing, in the sense that into such small peptides and entire essence is lost. Like macrophages, they do. So they will completely degrade it. No presentation, nothing. Not that way. It will be degraded in a way so that peptides are generated. You know, 8-mer peptide, 20-mer peptide, different kind of peptides will be generated. Now, these peptides will be loaded onto these so-called M2C compartments or M2C molecules. And this MHC class 2, which is happening all the way here into these, the Golgi apparatus, will be loaded onto these vesicles, which you call it as a M2C vesicles. Now, these M2C vesicles will be loaded with these peptides, and these peptides will go and present it along with these M2C compartment. They will go and present it on the surface of your dendritic cells. So this is a peptide MHC class 2 molecule. Okay. Similar way, there is a MHC class 1 molecule where you have any of these endogenous peptides, like you will have viral infections, etc. So all the endogenous proteins, they will be processed via this proteosomal complex. They will be loaded onto this transporter of antigen complex, the TAP complex, and this will be loaded onto the MHC PEP1 class molecule, which gets then loaded and targeted onto the membrane. Okay, so this is the class 1. And merger between this classical class 2 and the class 1 is called as a cross presentation. So you have a classical class 1 mediated pathway, classical class 2 mediated pathway, and you have a cross presentation. So this is a way by which an antigen is presented. In a nutshell, it is very simple. Your peptide of the antigen, not the whole antigen, it will not be recognized. The peptide, the processed peptide, should be loaded onto the class 1 or the class 2 and will be shown up to the dendritic cells. So this is all in all the importance of the dendritic cells. Now, what I have done is, just for making it much simpler and very interesting, I have divided it into 2 parts. First, I hope we will be able to cover, because I will not get into too much of complexities, as you may not even know the basics of it. So let us, dissect it so that you at least get the basic. So now, see, this is the MHC class 2, and we will restrict ourselves to the MHC class 2 presentation. We'll not get into the class 1 or cross presentation. How? Now we know the endpoint. One endpoint we know that if you give a Salmonella bacteria to the mouse dies, right? That is the endpoint. That means something is going wrong. The dendritic cells are there, macrophages are there, immune system is there, but then something is going wrong because the mouse are dying. What is it that is going wrong in the dendritic cells is the question now that you need to pose. That is what Mayuri did to understand. So when we were doing my previous postdoc lab where I was a postdoc, a Humboldt fellow, they showed that the Salmonella somehow inhibit the antigen presentation. And it was not known why and how do they do that. So we wanted to understand that how do they do this. And you can do this with very simple experiments or experimental tool. Mayuri also mastered the technique of culturing of the dendritic cells. Now, Ralph Steinman not only discovered dendritic cells, but he realized that these cells are so fascinating that it will be futile if we do not make them to grow in culture and do long-term experiments. But remember, unlike macrophages, these dendritic cells are very difficult to grow. You need to grow it from the progenitor cells in the bone marrow. So you need to do a bone marrow culture and then give them a specific growth factor like GM-CSF and certain other interleukins to make them into dendritic cells. How do you know that they are dendritic cells? You have a specific marker. So CD11C is a characteristic marker for the dendritic cells. If you have an antibody and if you show that these are CD11C, you can close your eyes and say, yes, these are dendritic cells. And they are very pretty when you look them under the microscope. Very fine, beautiful dendrites. Okay. So yes, so the dendritic cell culture was established by Mayuri and she was very successful in getting this in culture. One of the most important tool if someone wants to do a very high-end research in antigen presentation, which is the core and the heart of everything. If you want to ask a question, why my vaccine is not working, you need to go back again to the dendritic cells to ask, what is the reason that it is not getting processed? That is why your responses are very feeble. Or is the dendritic cells getting tolerized? What is the reason? So you need to have that technique. So if you see here, these are nothing but the total MHC2. Now, because we are looking into the MHC2 mediated presentation, you also have an antibody to MHC2. So once you have your cells in hand, and then you have your bacteria in hand, STM is Salmonella typhimurium. Okay. PFA means you fix the Salmonella. So what happens when you fix it with paraformaldehyde? That means the Salmonella is dead. Now that the Salmonella is dead, you need to ask a question that whether my presentation is because of the live Salmonella or the surface molecules which are there, because after you fix it, the surface molecules will be intact. Okay. And what are the very important surface molecules, colleague, on a gram-negative bacteria? This you have studied. One of you quickly tell me. It is there in the slide. 1 minute. Hello. Yeah. I'm in a class now. Can I just call you later? Oh, thank you. So, can anybody tell me which is a characteristic molecule on the gram-negative bacteria?
Ma'am, lipopolysaccharide.
Lipopolysaccharide.
Very good. Lipopolysaccharide. It is not present in gram-positive. What is there in gram-positive? Lipoticoic acid. Anyway, good. So, lipopolysaccharide is one of the very important, you call it as a pattern. You can call it as a microbial associated molecular pattern or pathogen associated molecular pattern. And the LPS binds to the TLR4, toll-like receptor 4. Anyway, we'll not get into that. So LPS is intact on the surface of fixed bacteria. So this is very important control. Whenever you are working with a pathogen, make sure that you have your control as a dead pathogen or a non-active pathogen, metabolically inactive pathogen, but you need to keep your surface intact, right? So this is nothing but a facts analysis to show that when you take your dendritic cells, you leave it uninfected or you infect them with Salmonella live or dead Salmonella, what is the level of MHC? Okay. So you have the uninfected one, this is the green. you have some basal levels. So the shift, the MHC is antibody is tagged with a fluorophore, right? So how do you see it in the facts? What is facts? Fluorescent activated cell sorter, right? So the facts will count the amount of fluorescent. So if it is on this side, that means the amount is high. If it is on this side, that means the amount is low. So the uninfected one, the green one, you see that the basal level of MHC is quite high. And the PFA fixed dead bacteria is not changing the basal level. But look at this red bar, what it is doing, which is a live infected. DC infected with live bacteria. It is shifting. That means that it is doing something to the MHC. It is doing something to the level of MHC. So this is shifting. That much you should that much only you keep in mind, that the Salmonella downregulates the MHC, which is present in the cell. And so what is again to be done is to figure out that what is it that is downregulating. So LPS, I told you, and you also mentioned it is present in gram-negative bacteria. It is one of the most important pattern. And you get LPS from sigma in a pure form. You can buy LPS, you can do many experiments with a purified LPS. But remember, colleagues, once you use the purified LPS, never extrapolate your data to the whole bacteria. Because though LPS is a part of the bacteria, when they are in isolation, they simply behave differently. No questions asked. That is a very interesting part. Which bacteria, see, this STM, Typhimurium, has LPS, but this Typhimurium is downregulating, completely downregulating your MHC. Only LPS in pure form, it upregulates. So the signaling by pure LPS is very different from signaling by whole live bacteria. And when you give killed bacteria, the killed bacteria is not able to downregulate. Okay, it is not able to downregulate. This very clearly suggests that wild type or the pathogenic Salmonella is now playing around with one of the very important molecule of antigen presentation, that is MHC class 2, and it is downregulating it. It is just not true in the vitro, but also in the vivo. So what happens? What do you mean by in vivo? In vivo means that you are doing this experiment in animal. Okay, so mouse is our model system. So you take a mouse, you give them Salmonella, and then you take the spleen, right? Because in the spleen, you have the splenic dendritic cells, very good amount. And you isolate the spleen, stain them with CD11C so that it will only stain your dendritic cells. And then in that background, you stain it with MHC. Same thing is seen here. The moment you infect them, look at the shift, look at this shift. This is the wild type, yeah? And the shift, it is shifting towards this side. That means it is undergoing some kind of change. The MHC level is undergoing a downregulation. Confocal shows the same. So CD11C means it is dendritic cells. This STM infected, green is the Salmonella. And you see that these dendritic cells are there. You know, it is not that the dendritic cell by themselves are going away. No, there are good number of dendritic cells because these are MHC, CD11C. But look at the staining of the MHC molecule. Such a beautiful staining in the uninfected dendritic cells. And the moment you infect them, the level goes down, even in confocal. It's completely going down. Yeah. So you have these nice dendritic cells and you have very low MHC in these dendritic cells, very low MHC. That means that Salmonella downregulates the MHC2 expression in dendritic cells. I hope that this much is very clear to you, that it is very simple, right? That once you give infection, they are downregulating the expression. Then comes the question in your mind. Of course, it is downregulating. So the downregulation can be due to the decreased synthesis of MHC class 2, or it is not the synthesis, but the MHC is made, but it is degraded. You got it? So anytime, you see this up and down regulation of a protein, you need to ask yourself two questions. Is it at a transcript level that means my mRNA itself is downregulated. That is why I'm not getting the protein. Or there is no difference in the transcript, the mRNA, it is produced, but the protein is not stable. And there are ways that you prove these two hypotheses, right? You have to think very clearly, jot down your experiments and prove these hypotheses, okay? So this is a decreased synthesis, it can be, or increased degradation of the protein. It can be any way. How do you do that? First, exactly you do the RT or the real-time PCR of the transcript. And you see here that the MHC2, and you, of course, you will have to normalize it with a housekeeping chain, which is a GAPDH. And you see that under all conditions, don't bother about the condition, you give LPS or you give Salmonella for 2 hours, 6 hours, 20 hours, give them heat or the PFF fixed Salmonella for 2, 6, 20 hours. There is no difference, statistical difference in the transcript level. That means that the MHC2 transcript is quite okay. The MHC2 transcript is quite okay. But then that is not the reason. This is not the reason for the decreased synthesis. What is the reason? Is it increased degradation? So this only option remains. So we have to now figure out whether this is this option, which is remaining. So one of the most important pathway by which a protein is degraded is a ubiquitination mediated degradation. Okay, the so-called proteosomal mediated degradation, which will have a ubiquitin. So it will get it targeted towards the proteosomal mediated degradation. So now if you stain your cell lysate with antibody to this so-called, this is a polyubiquitination, or you do an IP, these are all the kind of experiments which will only tell you that it is indeed true that the STM or the infected one have more of this ubiquitin tag. Do not bother about this experiment, because each experiment is a technique. Each technique's another is, 40 minutes to make you figure out that what is this technique. So a couple of things you must know. What is meant by immunoprecipitation or the pull down? How do you do the immunoprecipitation in a lysate? Yeah. So this is a very important tools and techniques which you might have learned in techniques in immunology. So in a nutshell, the STM or the wild type bacteria indeed showed more of these ubiquitin tag or polyubiquitination or the ubiquitin tag for MHC. That means it looks like the MHC molecule itself is undergoing some kind of degradation because it is tagged with the ubiquitin molecule, which is not happening either in the uninfected one or the killed bacteria. The question is how? Because ubiquitination can be of many types, yeah. But the one which allows them to be undergoing degradation or tagging it to the degradation is lysine ubiquitination at K63. So K63 position, this particular lysine 63 ubiquitination in a protein marks that protein towards proteosomal mediated degradation, okay? Not the K48. That is very important for you to keep in mind. Remember, colleagues, each one of them that I am bubbling out has a big process in cell biology. For you to understand the different kind of ubiquitination, which is a kind of, you call it as a post-translational modification. One of the post-translational modification is ubiquitination. You have two different kinds of ubiquitination, K48, K63, and depending on the kind of ubiquitination you have, it is targeted towards a degradation pathway or towards any other pathway. So a small lesson for you, when you go home, just brush up certain basics of processes of ubiquitination. Then it will become much more clear. Now, here, what is very evident from all these plots that it is not the difference in the K48 mediated ubiquitination, but the moment Salmonella infects dendritic cells, the MHC class 2 molecule undergoes K63 tagged ubiquitination. And because it undergoes K63 tagged ubiquitination, it is undergoing degradation. And what happens when it undergoes degradation? There are no MHC class 2 molecule for the antigens to be loaded and taken up. And they are very beautifully surviving there without any problem because they are not getting presented. They are simply not getting presented. And that is how they are inhibiting the antigen presentation. And very amazing, wonderful strategy. The same kind, but in a different way, is even done by mycobacteria and most probably by many other pathogens who are very successful. So Salmonella enhances the K63 linked ubiquitination of MHC class 2 in the dendritic cells. Now, this is also being confirmed by the confocal microscopy. And there are now many other ways now you have to be proving it. Of course, it is undergoing ubiquitination. Where is it undergoing ubiquitination? Is it undergoing endosomal ubiquitination, or it is undergoing a proteosomal ubiquitination? So all these experiments which are here just tell you one single thing, that once your MHC class 2 is there and it sees the bacteria or the Salmonella typhimurium, the Salmonella typhimurium does something to the system which makes the MHC class 2 ubiquitinated in a pH dependent manner. Okay. Now, all of these chemicals, you don't have to worry. You know, these are all the beautiful experiments to show that if, see, look at this bar, only Salmonella. You do not have any MHC here. This is a MHC, presence of MHC. You don't have any MHC. Look at this down bar. Compare it with UI, which is uninfected. But the moment under the same infected conditions, you add a chemical agent which will enhance the acidity, stop the acidification, like polymycin or bafilomycin. These are VATPase inhibitors. Okay, these are the chemicals which will inhibit the acidification. So the moment you inhibit the acidification, you see that the MHC levels are getting restored, not to the level of uninfected, but yes, it is getting restored. That means that the Salmonella is very intelligently maintaining the so-called acidic pH by recruiting certain VATPase, thereby leading to the downregulation of MHC by tagging it to the K63 ubiquitination. That is the entire hallmark of this study. All the rest of the corollary experiments were done just to prove this one single point, that how and why Salmonella ends up in degrading MHC class 2. Okay, so the internalized MHC class 2, what happens is it undergoes a proteolysis in the endosomal compartment. Because the endosomal compartment is acidic in nature and Salmonella makes it so, so it undergoes a proteolysis and thereby it gets to the evasion mechanism. Now look at this, immature dendritic cells. What is immature dendritic cells? The cells which have not seen the antigen. Okay, these are immature dendritic cells. Though it is immature dendritic cells, but that means not only the antigen, by the way, it is also very important that it gets some stimulus, like cytokine stimulus or other co-stimulatory stimulus to get it mature in the mature form. Now, when the dendritic cells is in the immature state, you see that these certain component like March1, this is one of the transcription factor, and this ubiquitination process, etc., all of them, they will make these molecules degrade in a lysosome. That is the immature dendritic cells status. The moment the dendritic cells are activated with the help of an antigen or with the help of any cytokine, these entire this particular transcription factor, it separates and it does not now allow your antigen to be ubiquitinated. So there is no more suppression here. It does not fuse with this lysosome and does not undergo, a degradation. So this is a mature dendritic cells, the work of the mature dendritic cells in a nutshell. So now what is the status of the March1? Now, if March1 is such an important transcription factor, what happens is the moment your STM or the dendritic cells are infected, this transcription factor level itself goes high. And it goes high in such a way at a very nice time period, you see, 2 and 6 hour, early time point of infection, where all the processes should be done. Not at very late time point, 20 hours, everything is done. So you don't need any March1. At this particular time point, the March1 is upregulated. Why? Because the moment it is upregulated, it is going to make the MHC polyubiquitinated and degrade. Okay, and this is not done by the killed bacteria. That means it is just not the job of the lipopolysaccharide which is doing. You indeed need a live bacteria to bring about these changes. It's a very active process. And along with that, the cytokine called IL-10, which is one of the most important determining cytokine secreted by the bacteria, by the dendritic cells, also goes up at 2 hours and 6 hours. So all in totality brings about the March1 expression, and this particular expression may be in a IL-10 dependent manner. So everything that you try to delineate one step of the other, immunology is so complex that you will just open up like a Pandora's box over and over and over again. So you will open it up like Pandora's box. Now, this Pandora's box I will not open because this will make you much more confused. But what I will only tell you, instead of getting into all the confusion, I will come to the end of this. And then we will leave it out for more questions because it's now 11:48 and we have good 10 to 15 minutes for question. When there is an infection, that means the dendritic cells seize the bacteria. These bacteria are taken up by the dendritic cells, and the bacteria, instead of getting its antigen presented, what it does, that it is getting slowly presented, but it gets this MHC molecule, see this MHC molecule ubiquitinated. And the March1 is upregulated, which leads to much more ubiquitination of at a K63 position. Okay. And when this is ubiquitinated, this is targeted to degradation. And if you do not have this live bacteria, then it cannot do this process and it undergoes presentation. Okay, so along with this, this also upregulates the IL-10. Now, IL-10 upregulation, proteosomal degradation, and inhibition, ultimately the inhibition of the antigen presentation, because there are no MHC2 compartments or there are no MHC2 molecules left for it to carry it to the surface of the dendritic cells and show them to the T cells. Okay, so this is how it degrades, or this is how it inhibits. Salmonella successfully inhibits the antigen presentation. And no doubt that they are so smart that our body system cannot take care of this pathogen. So we do need an intervention strategy in this. Okay, colleagues? So now what we do is we open it up for discussion. Please ask.
Ma'am, yes, ma'am.
Yes, each and every
Yes,
tiny questions you ask me. Okay, ask me all.
Ma'am, the thing.
Yes, go ahead. Go ahead.
Ma'am, the thing is that, ma'am, you are from IISc Bangalore and I am not able to understand. Ma'am, I am from NIT. So, ma'am, technological aspect, ma'am, after 2 years our college has opened, ma'am, I need very, very sharp, minute details. So, ma'am, can you arrange any from IIT Bombay professor, because Anil Gupta sir has told that please raise this forward, that we should get tiny, minute details. Each and every detail should be taught in the class. So that only I think I want.
Tushar, what you need to do is, see, say for example, I do take all the classes in JNC. I teach in Kalyani University. You need to take it up to your mentor or your sir or your teacher, and then tell them to organize a lecture series of immunology. Okay, so either I do not know, that may not be possible that you get integrated in our own course in immunology, which is going to happen soon, but that I teach a host pathogen interaction, that may not happen. So I need to chip in to really teach you this minute detail. Okay, so bridge between your own mentor, and then let us see how we can get into this very minute detail. And this detail comes from your developmental immunology. How do you first of all get these dendritic cells? What is meant by T cells? What is meant by thymic education? What is meant by B cells? What is meant by tolerance? How the dendritic cells can process? What are these MHCs, molecules? How are they themselves processed? What is the difference between class 1 and class 2? How are the antigens processed? Why there is a certain peptide of this length and not the other? You got it, Tushar? So these itself
Yes, ma'am.
counts. So you kindly get back to Professor Gupta and figure out how do you do this. Yeah.
Yes, ma'am. Yes, ma'am. Okay.
Okay. Kindly make sure to do that. And then what you need to do is you need to do a lot of self-reading. See, if you are really interested, you will try to do so much of self-reading. So kindly read QB's Immunology. I'll show you. This is QB's. I don't have now with me. This is the book by Janway. You must. Janway's Immunology. It's extremely, extremely important for all of you to read, to understand.
Yes.
Right from the
Yes, ma'am.
Susumu Tonegawa's, start from the Susumu Tonegawa's Nobel laureates BDJ recombination, which gives you the beautiful glimpse of the diversity of the B cell and the T cell, antibody diversity, you know, T cell receptor diversity. Start from there. Actually, you need to start right from the page 1 and try to understand. And once you do this homework, you will see that any lecture is becoming very easy for you. Very, very easy for you. Okay.
Yes, ma'am. Yes, ma'am. Okay.
Yeah, good. So now there are a few questions in the chat I will take. And in the meantime, colleagues, you also start asking me questions. Okay. It's we have some time now. Okay. Why sometimes this is question from Mahi Rathod. Why sometimes our immune system don't respond on time? I mean, sometimes it's very late for us and they infect many cells. Yes, Mahi, that is the reason that we call it as a, evasion strategy. Why sometimes our immune system do not respond in time? The system which responds in time, by definition, should be always there. There should not be any production. That means there will be a delay. That system is known as an innate immune system. Dendritic cells are one of the most important cells of the innate immune system. And in the innate immune system, what are something which is already there like a gateway, like soldiers? First of all, apart from your physical barriers, what do you have? You have these called as a toll-like receptors. Now, these receptors are already there on the surface. They will immediately sense the pathogen and they will start the cascade. During this cascade, actually, the cell defense system is building up. But there is something that what I told you, a small example of how they evade. Because the arms race is beginning. So the pathogen will have its evasion strategy. And that is the reason it is late. And of course, they are smarter, they are faster, they are smaller. So some cells, some bacteria like Salmonella or Shigella, they replicate much faster. It's not that every bacteria does so. Now, your second question is if a person is suffering from tuberculosis, we don't know in the beginning. We come to know only when it's disease. Because why? Tell me why. Tuberculosis, Mycobacterium tuberculosis is such an amazing pathogen or a bacterium that its division time itself is very slow. Unlike Salmonella or E. coli, which divides in the broth every 20 minutes, Mycobacterium will only do it once in 14 days. 14 days. So when they go in the lung, they are cool. They just go there. Nicely sit. If the body system is very good, they are going to have it very fast. Eradicate. So they are cool. Once in 14 days, slowly they will replicate. And that is the reason it takes years together to manifest the tuberculosis. Got it? So it is the beauty of the pathogen, how much of the arms race that it can deal with. It's an arms race, the body is triggered, then it attacks. So once this attack comes, this will not keep quiet. This will also attack. So it's rare that we get a very beautiful story of endosymbiont, from where your mitochondria came, our mitochondria came, or the chloroplast came, that one of the alpha proteobacteria was engulfed by some other bacteria and it became a beautiful cell. It's no longer the case. It's because of the arms race. That is the reason. Okay. Okay. So what is March 1? It has a very big name. Sorry. If I want to describe, it is a transcription factor. Simply say that it is a transcription factor. You can look it up. It is one of the very important determinant for understanding the process of its triggering certain genes, which will again lead for the ubiquitination. Okay. So how Salmonella infection induce March1 expression in the infected? Yeah, good question. Very good question, Rashi. Now, Salmonella is a vacuolar pathogen and it has many of the effector proteins. We are trying to understand what are these effector proteins. It's a very tough job. You know, it has a secretion system and it can secrete 40, 50 odd effector protein. It can be a combinatorial effect of those effector proteins. So we are trying to understand that what are the effector proteins. It's difficult to dissect. It may not be one. It might be 3, 4, something. So yeah, so if that is dissected, then yes, you say that these are the effector protein which lead to the upregulation of the MARC1 and then the upregulation of the K63 mediated ubiquitination. Ma'am, in MyD88 dependent signaling, why only, 1 minute, why only TLR3 is not use this pathway, but all other TLRs use? Okay. Why did you only say TLR3? MyD88 pathway can be MyD88. What is MyD88? MyD88 is an adapter protein. Okay. So a lot of these toll-like receptors, they recruit the MyD88 and then they transduce the signaling. Now, if you know a bit about toll-like receptors, there are toll-like receptors which are present on the surface, that is on the plasma membrane. You have TLR1, TLR2, TLR4, TLR5. There are 3 toll-like receptors which are present on the endosomal membrane. What are they? TLR3, TLR7, TLR8, TLR9. Okay, TLR8 is not one, TLR9. So TLR3 is endosomal toll-like receptor. So it takes another adapter molecule to recruit or to signal the processes. Got it? Yeah. So MyD88 is the one which is recruited onto the tolls which are present on the plasma membrane. And in that also, TLR4 itself, which is present onto the plasma membrane, undergoes 2 kinds of signaling. One is MyD88 dependent, one is MyD88 independent signal. Right? Okay. So, anything else? Anything else? Tuberculosis is done. Yeah. So what is the take-home message? How do you take care of your immune system? The first very important take-home message here is that the immune system is a very complicated, alarming system, okay, in us. And there is a strong correlation of your second organ, that is a gut flora, mucosal maturation, and your immune system. So the first thing you need to do is kindly do not play around with your gut flora. Kindly try to maintain the diversity by not overdoing your habits, food habits, this habit, that habit. And you will see that to some extent you're indeed protected from the normal day-to-day infection. You will not come down all the time with cough, cold, these, that, you know, diarrhea, dysentery all the time. No, you will be very safe if you take care of. Secondly, once there is a pathogen attack, because the pathogens have so many different strategies, it becomes very difficult to even answer the question that how can you, until and unless you have an antibiotic intervention therapy. Now that also is not okay because you are giving it a threat to the system. Okay, so most importantly, do not allow it to get inside. At least foodborne pathogens like Salmonella, Shigella, Listeria, certain viruses. You can always take care by not indulging on the food which is not cooked, always washing your hand, etc., etc. So that part is taken care of. Always drinking boiled water, not drinking water from anywhere. So all these precautions you can take. When it comes to aerosol infections, you know how dangerous we are because we are in the 3rd year of COVID, and you know, right? So air is everywhere. If I get it, if it is now COVID is here, I will inhale. And we did that for 3 years. We were with mask. Now also I am with triple mask. So anyway, so you can, there is something in your own capacity you can take care of yourself from infectious disease as much as possible. Please take care, because once it is in, it is very difficult to get rid of it. Okay, colleagues, thank you very much. So my time is up. Yeah, please reach out to me. Anything that I can do, I do take too many classes just to make one point clear, that you need to understand the subject. Then only you are going to have that love for the subject. Okay, so please understand it. If you cannot, I will help you in whichever way it is possible. Thank you. Thank you, Dr. Murugan.
Thank you, Professor, so much.
Yeah. So take care and bye-bye.
Students, we will wait for our next guest to join us. Please stay with us. Okay. Oh, doctor, you have joined.
Yes, I have joined. Yes.
Thank you. Okay, students, we will continue with our next lecture for the day. We have with us Dr. Pranab Nanda Bhattacharya. He's presently working as an assistant professor in NNS College, Titabar, Dilbarg University, Assam, India. Earlier, he was scientist C in Mycology and Microbiology Department, Tea Research Association, Jogat, Assam. He was a research fellow postdoctor at ICAR during 2012. He has academic experience as lectures at the two provisionalized college of Assam, two-time gold medalist at graduation and post-graduation in botany from Gauhati University, obtained PhD from Gauhati University, Assam, specializing microbial ecology and bioprospecting. Areas of present research include microbial ecology, bioprospecting, metagenomics, soil microbiology, agriculture microbiology, behavior of microbes, plants, pathogen under change in climate, plant microbe interactions, and microbial mapping. Also adoption of non-chemical and organic approaches in agriculture, soil restoration, etc. He has received awards and honors like National Merit Scholarship, Gold Medalist from universities, Professor A.C. Datta Memorial Award, Best Researcher Award from International Scientist Award 2020 on Engineering, Science and Medicine by prestigious V.D. Good Professional Association, India. Sadhana Young Achiever Award, BioWed Young Scientist Award, Annual Member of the Association of Microbiologists of India, AMI. Recently joined as a project coordinator with the esteemed International Asia Pacific Network, CMBP, for developing research projects for promoting agroecological practice for improved soil health and productivity. He has published more than 50 number of research papers in national and international journals. He has published 2 books entitled Phyllophane Microflora in certain tea cultivars of Assam, Northeast India, and exploiting their potential use in tea, and actinorhizal plants, Elagnus latifolia, conservation, perspectives in reclamation of degraded land, including tea soil, with International LAP German. Participated in a number of national and international conferences, seminars, symposiums, and presented own research area of research. Has guided a number of undergraduate students from different academic organizations throughout India. And recently, a number of interdisciplinary research works in progress for technological dissemination and extension activities in relation to tea consortium. He also reviews a number of journals and other publications. So with this short introduction, I invite Professor Pranab to deliver his lecture. Professor.
Yes. Thank you, Dr. Murugan, scientist and Biotechnology at this program. So thank you for your nice introduction on me. So now I think without wasting our time, we should go for our presentation because we have also some stringent time frame. Yes. So my dear students, so today's presentation will be on basically on use of microbes. Okay. So use of sustainable approaches in agriculture, how we can use them in agriculture for betterment of our agricultural productivity. And while presenting this, I will exemplify it with it as a tea as a role model ecosystem. Okay. So now let's move to our presentations. So now I want to share my screen. Dr. Murugan, is it okay?
Yes, Professor.
Hope it is visible. No? I think it is visible.
Yes, Professor, visible. Yes.
Yes, it is visible. So,
Yes.
so students, now we move to our presentations. So potential means exploiting the use of microbial resources, basically the decomposers, okay? So for their benefit or for their exploitation in decomposition of the agricultural waste is our today's topic. So, okay. Is not moving actually. Okay, it is moving. It is moving.
Yes, Professor, it is moving.
Yes,
Your
yes.
voice is little low. Can you
Yes,
put
yes,
mic
yes.
closer?
Yes. So now you all know that agriculture. So agriculture basically requires nutrients. So we all know that nutrients are needed for betterment of plant growth and development. We have 17 essential nutrients. So out of these nutrients, some nutrients can be considered as a macro elements or macronutrients. Some of them are certain secondary nutrients, and then some of them are some micronutrients or trace elements, which are also required for normal growth, development, and reproduction of the plant. So without the assistance of these nutrients, we all know that plant growth and development and reproduction and entire metabolism activities will not go properly. So therefore, nutrients are needed for each and every corners for agricultural productivity as well for developing the plants. Okay. So now these nutrients, we know that they are essential for lot number of actually functions. Likewise, suppose nitrogen is necessary for protein formation, nucleic acid formation. Then phosphorus is required for nucleic acid formations, then ATP synthesis, phospholipids. Likewise, potassium is also required for different vital activities like enzyme activation activities. In today's discussion, as we will discuss about that, means delivery of some cellulose decomposing microorganisms. These microorganisms are also known for the production of some essential microbial categories like cellulose. So this type of enzymes plays an important role. Okay. Then sulfur, this is also another macronutrients or the nutrient which is required for means growing plants in larger amounts. So these macronutrients, likewise the micronutrients, they all are basically actually required for normal plant growth and development. And we know that mass flow, division, interception, these are some of the technologies by means of which these nutrients are getting absorbed by our roots. Because we all know that plants are rooted in soil, and roots are the only they are organs to get the nutrients from soil. And while they got their actually some of the nutrients also from atmosphere. So you all know that photosynthesis is the vital process in each and every living organisms, and it is the basically run by the photosynthetic plants. Okay. So we know that chloroplast is in there, then chlorophyll molecules, they play an important role while bringing together this carbon dioxide and water molecules. Water molecules is absorbed from the by means of roots, and then carbon dioxide is getting absorbed from the atmosphere. So as a result of which this entire process of photosynthesis runs, and the importance of this photosynthesis lies in the production of basically, you all know that is the glucose, that is the means food, ultimate food, and the entire living world, it is dependent on this food. Without this production of the food, the entire living world will not to be sustained. So that means what? So these nutrients are required for normal plant growth and development. Besides, as we heterotrophic organisms are also dependent on plants, so we also require nutrients. Okay. So now we all know, as nutrients are required for the plants, so there are also some problems due to their deficiency. Okay. So nutrients are required for the plants for normal growth and development. However, due to the deficiency of these nutrients, so there are some problems appeared. So as we all know that these are problems like yellowing of the leaves, then yellowing and brown leaf edges, okay, and then holes in the leaves. There are some necrosis, chlorosis, yellowing between the leaf veins, small and stunted leaves, then curled leaves, yellowing. Then all these types of these are basically they are related to nutrient deficiency condition. So I will mention you why nutrient deficiency actually causes in soil, because you all know that soils are entirely rich with nutrients actually, but how this deficiency occurs? So this is the actually reason why we should go for application of some fertilizers, because as soil is naturally rich with rich in different kinds of nutrients. They may be either nitrogenous nutrients, phosphorus nutrients. But what is happening due to cultivation, continuous cultivation? Likewise, in case of suppose tea ecosystem, if you assume tea is a monocropping agricultural practice, no? So lots of you have to plant the tree for so many years, up to 50-60 years. So productivity runs. So likewise, in case of agriculture also, there are lots of agricultural plants we planted in our agricultural fields. As a result of which the plant require nutrients from soil. And ultimately what is happening is the soils are also depleting the nutrients. So this is the reason we have to refill the soil nutrients. And farmers regularly use those fertilizers, okay? Or fertilizers are also needed for improving the status of soil. So it can be defined likewise, the fertilizers are simply nothing but plant nutrients, as we supposed to macronutrients, micronutrients, and some of the trace elements. So all of them are actually required for normal plant growth and development and their functioning activities. Okay, so now if we go for fertilizers, so these fertilizers are also of different types. So they may be either natural fertilizers or they may be either some chemical fertilizers. So natural fertilizers, you all know, these are basically the organic resources. So natural fertilizers, they may be either soil amendments. Soil amendments are nothing but these are basically added to soil to improve the quality of soil and as a result of to improvise the plant growth properties. Okay, plant growth properties means leaf number, leaf size, stem girth, rootlet size. Okay, so these type of things they improve. So likewise, these soil amendments, they may be either biosolids, they may be either biosalts. Okay, so these are basically organic resources. Another thing is the organic biofertilizer, which is today's concern. So we have to improve the sustainability in our agricultural system. Okay, so without sustainability, our agriculture cannot run. That means sustainable approaches. What does it mean? It is basically nothing but it should be a cost-effective approach. It should be a non-conventional approach. It should be a non-chemical approach, and you can use the benefits of organic biofertilizers like your manures, compost, then animal excreta. So these are basically organic substances. And another chemical means source of fertilizers is they are chemical or synthetic in origin. You all know urea, ammonium sulfate. Okay, ammonium means nitrate, rock phosphate, superphosphate. These are different examples of nitrogenous fertilizers, phosphoric fertilizers, or potassic fertilizers. Okay, so these are the chemical fertilizers. So now what is happening due to the use of these long-term applications of these chemical fertilizers, our soil is getting worsened day by day. So why? How we can improve our soil now? So this is the strategy improvement. Okay, we have to improve the status of soil. Without improving the status of soil, we cannot improve the properties of plants or for their growth promoting abilities. And besides, the plants also getting in stress, okay, due to rapid applications of chemicals. So now, likewise, pesticides are also different types. They may be either basically two types. So it may be either organic or it may be either inorganic. Inorganic is again chemical pesticides. There are a lot number of chemical pesticides, and these pesticides are usually added in agriculture for plant protection purposes. Plant protection, you all know that normally there are some native conditions of the agricultural plants. Suppose in case of tea plants, so northeastern tea plantation, suppose northeastern tea plantation is basically a humid condition, rain-fed condition, and this rain-fed condition is usually helpful for the establishment of different kinds of diverse kinds of pathogens, and pesticides are regularly being used for controlling of these pests. But what is happening due to continuous use of these pesticides, the pests are also getting resistant day by day. So now any kind of agriculture also this happens actually. So therefore we have to go for reduced or limited use of these pesticides, and we have to bring back our soil with the application of some biological pesticides. And these biological pesticides, this is microbial pesticides. And this, we all know that these are the harmful effects of pesticides. So there are lots of disadvantages while using these chemical pesticides in soil. So chemical pesticides, with the application of chemical pesticides, it causes air and groundwater pollution. Pesticides are normally added to soil. Okay, so it gets runway to the nearby water bodies, and our natural events, some means happenings. There are some natural happenings like our vaporizations, then evaporation activities. Likewise, they again get back to our soil conditions. So ultimately air and groundwater pollution happens. Soil contamination, this is one of the important problems. So soil contamination is related to chemical toxicity. Then reduces biodiversity, including microbial resources. So microbial resources, we all know that soil microbial resources or rhizosphere microbial resources, they have a very immense influence to improve the plant microbe associations in soil, and ultimately they help in plant growth promotion and making the plant more resistant to diverse pests and pathogens. So while application of the chemicals, they are getting deteriorating the rhizosphere environment. Then reduces nitrogen fixation and other essential nutrients. Then pollinator decline, whenever there are chemical toxicity, pollinators will die. Then destroys habitat, especially for birds, threatens endangered species, then chemical resistance development. So these are all harmful effects related to pesticides. So this we all know that this is the pesticide cycle. So how they get entangled with the atmosphere and the soil. So we are applying soil pesticides, they are getting contaminated in the atmosphere, again they get back to our soil conditions. So ultimately what is happening? Our natural ecosystem is getting ruined day by day due to rapid and, or you can call it as a illegitimate or exogenous applications of chemical pesticides. So these chemical pesticides, what they are causing, they are basically chemical toxicity is basically it can be exist in three forms. That is persistence, bioaccumulation, and then biological magnifications. Persistence means the chemical pesticides, whenever you use in the soil conditions or other any kind of environment conditions, they persist for a long time without any degradation. Then bioaccumulation means whenever it entered in our food chain, and biological magnifications whenever it gets magnified to our different food webs. Ultimately, it is getting within our body, within our tissues, within our entire environment, and our entire food chain and food air getting degradation day by day. So we know global warming. So pesticides, one of the important means implications of pesticides is the global warming, or pesticides are resulted due to or pesticides are causing global warming or warmer climate. Now you see in every corners of your world that there are issues of global warming, and you know that whenever chemical pesticides are actually, or chemical supplements are prepared or produced or manufactured, basically three compounds are released: carbon dioxide, nitrogen oxide, and then methane, and they are getting polluting our environment. So thus pesticides are also related to cause our that is toxicity in our environment or global warming. So likewise, if you assume the tea ecosystem, so tea ecosystem, there are three major problems: soil compaction, then erosion, and land degradation activities. So soil compaction is basically, it is one of the it is one of the most important problem in relation to pesticide applications or chemical applications. Then soil degradation, as a result of which soil is getting degraded day by day. And if our soil is getting degraded day by day, can we bring our agriculture back? No, because our agriculture will be in three: soil salinity problems, localized waterlogging problems in natural our tea growing areas. Whenever there are localized waterlogging problems, soil porosity is getting actually affected. Then fertility loss, plant productivity is getting lost. Predators and natural enemies are getting affected by this application of this chemical toxicity. Then contamination of our water bodies, nearby water bodies. The water bodies are regularly used by their gardeners or for other purposes also after purification. Then plants getting in stress. So this is one of the important problems, because whenever we regularly use or we illegitimate application of chemical pesticides, what is happening? Plants regularly getting in stress. And stress plants, as we all know, whenever we are also in stress, we also develop some kind of disease, because our body is more vulnerable to some pathogens or germs. Okay. Likewise, whenever the plants are getting in stress, they are not getting properly the nutrients, their soil is very hard. So like in that conditions, the plants regularly get in stress. And due to these stress conditions, lots numbers of insects or insect pests have been reported in the tea plantations. Okay. So they may be either different types, they may be either they may be affecting actually their foliage, means leaf portions, they may be affecting their apical portions, then stem portions, their root portions. Okay. So termites, suppose. So whenever there are termite infestations takes place, entire root becomes very weak and then it is getting died. Okay, so similarly, there is another problem that is one of the important major problem, that is key diseases conditions. Whenever the plants getting in stress, okay, so there is every possibility of development of a disease in a more severe conditions. Okay, so normally in case of tea, there are basically two types of diseases, primary and then secondary. Primary diseases basically occurs due to direct means infection by the pathogen, while in case of secondary disease development, predisposing factor means whenever there are some problems in related to stress, like soil salinity conditions, like core erosion problems, like pH problems. So these type of problems generally means getting the plants more stressed, and as a result of which the pathogens also getting means rapidly attack to the means plants. So thus there is a very need for sustainability development in our agricultural sector, and without the sustainability development, we cannot run our agriculture, because this is the most effective non-chemical and approach, because we all know, we have already learned the harmful effects of chemical supplements or pesticides. Okay, so it has also been mentioned by United Nations. You know that United Nations 2015, they have launched the what, that is the your means these are some of the means have used the sustainability issues, and the sustainability issues has been mentioned clearly in 8 different sectors. So where we have to use or different kinds of non-conventional approaches, like the application of organic practices, sustainable practices to reduce the limit of what, chemical supplements in agriculture or our natural systems. So agricultural sustainability improvement, agricultural sustainability by which way? Organic manure production. We can develop a process for the preparation of organic manure. We have organic manures or we have vermicompost, but how we can give it as a value added? Okay, so this is the strategy today, because until and unless value addition or until and unless quality compost preparation, our agriculture will not be sustainable. So for this reason, there are some important microorganisms in our natural systems which need to be isolated, which need to be characterized, which need to be utilized properly with some other resources like worm species. We have worm species, we have some effective worm species or efficient worm species, or these microorganisms and worm species, whenever they get in combined way, we can get a very quality manure production. Okay, so kinetics in organic manure decomposition using it is a very eco-friendly approach. It is a sustainable approach, means it is non-harmful and it is cost-effective also, and we can reduce the load of toxic supplements in agriculture. So this is the main motto, or this is the main goal while experimenting these things in our agricultural systems. So you know that lots of agricultural wastages are here, or lots of agricultural substances are deposited day by day in every corner of our world. So it can be used properly. If we can use properly with the addition of some our biological resources, likewise I have mentioned the cellulose degrading microorganisms. These microorganisms are known for the production of cellulose enzymes, so they can degrade these things to quality organic manure. And this quality organic manure, if we apply in our agricultural system like soil, reducing the compactness of soil, so very effective approach in integrated pest management and integrated disease management. We have rice straw in our ecosystem, we have waste safety loops. So with biomass, we have run some experiments actually basically on this use of these cellulose degrading microorganisms. We have collected this type of agricultural wastages, and we have, these are some of the photographs also from actually website also. We have also some original findings also, I will explore it. So these cellulose degrading microorganisms, how they can work? So these cellulose degrading microorganisms, I have already mentioned, they have cellulose degrading, means cellulose enzymes, and ultimately they can play an important role while degrading this means agricultural wastages to quality by fertilizer productions. So there are lots of, they may be either bacteria, they may be either fungus, or they may be either actinomyces. While in our experiments, we have also developed the process of formation of microbial consortium. So microbial consortium is nothing but this is that it may be a bacterium, it may be a fungus, it may be actinomyces in association actually. So whenever they are actually used as an association, several parameters need to be actually taken care of, because microbial, in a microbial consortium, all the microorganisms should work together. Okay, so one microorganism should not hamper the growth of other microorganisms. Okay, so likewise we have standardized some processes. So Bacillus species, Pseudomonas species, Serraceous species, Trichoderma, they are known for their effectiveness or their abilities in cellulose degradation or agricultural wastages conversion to quality organic manure productions. So these are basically their plate cultures. So whenever we isolate the microorganisms from soil or from any kind of natural environment, we can also collect, or we can also isolate from cow dung, okay, cellulose degrading microorganisms, then decomposed wood. So whenever they are practiced for their confirmatory test, so they can actually produce some halogens, and these halogens are nothing but these are their cellulose activities present. Okay, so we have done some experiments where we isolated and screened some of the cellulose degrading microorganisms, and these cellulose degrading microorganisms have been isolated from using different media. They have some specific media, and after isolation, another important thing is that whenever we are using some beneficial microorganisms in agricultural perspectives, we have to do some pathogenicity screening, because if the microorganisms have some pathogenic effects, or if they are pathogens, they should not be applied in the agriculture. So normal pathogenicity screening microorganisms, so they can be easily screened by using methylene 2-agar and then macron media. These media are basically colorful, and if our microorganisms are not producing some kind of they are selective media. No? These are selective media, so they are not producing their colors in those selective media. That means they are non-pathogenic or non-harmful. Okay, so this is one of the important testing which is need to be carried out while working with cellulose degraders. So this is the, you can see FPC. So here are two. This is the control experiment where actually blotting papers or workman filter papers have been used. This is the source of cellulose. No? So our microorganisms, when we have used, so in your right side you can see ZHLM. So they are getting degraded. The filter papers getting degraded, and in the control experiment it is as it is. That means it is not degradation, because there is no cellulose degrader. So that means this cellulose degradation microorganisms, they have the abilities or immense abilities to degrade the cellulosic contents in our agricultural wastages. So this is one of the experimental findings where we are getting some of the potent microorganisms for their effectiveness in our lab conditions. So molecular characterization is also needed for their identification purposes. So this is another important steps need to be carried out while working with cellulose degrading microorganisms for their bioconversion experiments or for their biotransformation experiments in agricultural wastages for the production of quality organic manure. So another thing, while using these cellulose degrading microorganisms for the production of quality organic manure, what do we mean by quality? What do we mean by quality organic manure? That means it should be rich in organic nutrients. It should be rich in some beneficial microbial categories. It should be rich in some antagonistic microorganisms. This is another value addition for the production of this quality organic manure. Whenever we are using this quality organic manure, so we can also, again, there is another assets, or there is another possibility for the isolation of some phosphate-solubilizing microorganisms, nitrogen-fixing microorganisms, then actinomyces, antagonistic actinomyces. They can be again tried again in agricultural plants. I will show some slides where our microorganisms are also effective while in the management of some diseases in tea. Okay, so vesicular mycorrhiza we can isolate from this compost. Okay, so and the quality of these microorganisms or the beneficial category of microorganisms, the recovery patterns is also found to be very high than the normal soil. That means in the same edition of the production of this quality organic manure, we are also eventually able to isolate some of the other beneficial categories of microorganisms, which we can also apply in our agricultural system. So we are using these microorganisms for the callus formation, then rootlet formation. So further, while for experimentation purposes, we are getting that while applying this organic manure which is prepared due to these biotransformation experiments, so the more amount of callus will form, or the quality of the callus is very good, and then root formation is also very good. Then we have done some experiments in the pot cultures also, in the field conditions also. So these are some of the TB clones. So means T vegetative clones where we are applying this compost and we are getting a very good result. That means they are enhancing the soil conditions, natural soil conditions. With the compost, they are getting more, means microbial fortification is happening here. Okay, so these are some of our experimental plots where we are using this cellulose microorganisms in our nearby means experimental station, supply the experimental station. So here we are getting this mass culture and we are applying this in the field application. Okay, so as you know, we have come to know from our presentation that toxic chemicals. So these toxic chemicals are regularly ruining or regularly they are getting means deteriorations in the physical, chemical, and biological quality of our cultivable land. So while seeing this or while observing these things or while experimenting these things, so bioconversion of agricultural waste using the cellulose microorganisms in the agriculture sector is a good strategy for development of concept of organic practices in agriculture. So this is another experiment where we are getting a very good compost while using tea wastes. So tea wastes they can be decomposed. So you can see the color differentiation in control where we are not getting the composting coloration. So it is actually in a composting. After 15 days of composting, with the application of our microorganism, effective or efficient microorganism, we are getting a very good quality from compost. Okay. So these CTMs, why they are basically known for their degradation abilities. So, or why we have to use this cellulose microorganisms. These microorganisms, they have a rapid, as you all know, that microorganisms have some common characteristics because they are rapid in their growth and multiplication abilities. So they can be easily adapted to the conditions. Then expression of multi-enzyme complexes. Then ability to colonize diverse environmental niches. You can isolate the microorganisms from, I have already mentioned that you can isolate the microorganisms from cow dung, from soil, from any kind of your decomposed wood. Okay, so they are versatile in their mode of distribution or habitat. Then ability to withstand varieties of the environmental stress conditions. Okay, so these microorganisms now they are being utilized in the feed sector for the use of their efficient nutrient management practices and then plant improvement and for disease protection, integrated disease management practices. So along with this, we can also use the efficient worm population. So we have worm populations like Pesinia foetida, Paranox, Emainthus, then Davidia. So these type of worms, so they have a good potentiality to degrade the organic matter. So enormous prospects in using CDMs for agro-waste bioconversion. Microbial fortification is usually added for soil health, crop productivity, and eco-parameter. Then additional enrichment of the compost can be done with the application of some humic acids, amino acids, and minerals. That means after the production of this quality compost, you can also add humic acids, amino acids to it for value-added compost. Then potential or potent decomposer. These are worm species, efficient worm species. These are the potent decomposer of organic materials. So it can be used along with the CDM consortium or CDM inoculum for the preparation of organic nutrients or organic biofertilizers. So in one of the experiments, we have used a very low-cost production technology of what? That is biological fertilizers. So with the use of our efficient worm species and then our cellulose microorganisms, and it has also worked well in the under hilly and under hilly ecosystems or under hilly agricultural ecosystems of even in Meghalaya. Okay, so this is one of the experimental results: recycling of plant biomass into quality organic manure using a low-cost technology. So quality organic manure indicators. So what are the best indicators or what are the indicators for quality organic manure? So whenever we are preparing the organic manure, it should be quality. So that's why we are using cellulose microorganisms. So we can see the quality of the organic manure by our visual observation. Okay, so continuous means rotating or continuous means handling of this compost is required while minimize the their temperature, because whenever composting is done normally temperature is getting rise. So whenever we are rotating our composting substances, it is actually neutralized. Then moisture content is one of the important things. So good compost should be tested by a squeeze ball test. So in case of squeeze ball test, what is happening? Basically, three types of soils we can identify. If the soil forms a sticky ball, it is a what? So clay soil. Then in case of soil which is not forming a ball in our hand, so this is actually not good for our agricultural purposes. And in case of what is happening, biological manure production technology, the biofertilizer which is prepared due to this technology, so this forms basically a ball within our hand. And it is another thing is that, so if we get a pressure to this ball, it becomes a what? It is piece of cake. That means it has a good water holding capacities. So these are some of the visual observations, or these are some of the very simple tests by means of which we can signify the quality of our compost, which is prepared due to our technologies. Okay. Then another thing is that there should be very less amount of heavy metals. Okay, there should be less amount of toxic metals. There and another important thing is that pathogen status. After producing these biofertilizers, using this means non-conventional or non-chemical approaches like application of these biological strategies, cellulose decomposers, we should do the pathogen status. Because I have already mentioned that whenever we use something in agricultural system, it should not be pathogenic. That means, and one of the important things is that after preparing this compost with this way, the compost itself has a very limited availability of this type of pathogens. Because due to composting activities, they actually assist the production of lots of beneficial microorganisms. And these beneficial microorganisms ultimately, they have enough mechanisms, cerebral production and antagonistic activities, they ultimately ruins the life of pathogens in compost. Okay. So what are their advantages? Why they should be used in agriculture instead of this, or in order to reduce the load of chemicals? Because we are all well acquainted with the disadvantages of chemical pesticides or chemical supplements in agricultural system. Okay. So what are those advantages? They play an important role while providing plant nutrients. We know plant nutrients, they may be other macronutrients, micronutrients, or they may be other some trace nutrients. Okay, so these nutrients are added by the application of these organic nutrients, or organic means biofertilizers. Then naturally rich in diverse nutrients. So after composting activities, you can also, I have mentioned that you can also give value added, suppose humic acids, you can add amino acids. So it can again have the abilities to promote its effectiveness while in agricultural sustainability, or agricultural means what production. Okay, then improve soil physical properties. So plays an important role in improving water holding capacity, soil porosity. Okay, this type of things which is gradually deteriorating due to application of illegitimate or injustice, no, or due to exagerous, or due to lots of applications of chemicals day after day or years after years. So then increase rootlet size. We have already learned this in our tea ecosystem also. Then protection from phytopathogens. Yes, whenever we are using this organic or this biofertilizer in our soil conditions, they naturally harbors lots of beneficial plant microbe interactions, and these beneficial microbial interactions in soil after addition of these organic biofertilizers, they ruins the activity of pathogens, or they reduces the load of naturally, or they reduces the load of toxic pathogens, okay, in soil. So then water relations plays an important role in improving water relations. Then drought resistance in all activities, all heavy metal tolerance, okay. Then these are also important role as carbon dioxide fertilizer. Carbon dioxide released during decomposition acts as a carbon dioxide fertilizer. So these are all the benefits of while using this biofertilizer in agricultural sector. So now another advantage thing is that so they can be well adopted as the integrated nutrient management practices. Integrated nutrient management practices is nothing but this is the good agricultural practices. Suppose whenever we are using our nutrient applications, we normally come in mind that we are using chemical supplements like urea, suppose. Okay, in case of chemical means fertilization. But in case of suppose biological fertilization, we are using suppose a microbial resources like trichoderma. But in case of integrated nutrient management practices, what is happening actually? You have to adopt a good agricultural practices to reduce the load of chemicals. So good agricultural practices means cultural practices. Then you have to adopt the biological practices with the recommended use of some plant protection formulation. So while developing these integrated nutrient management practices, similarly integrated disease management practices, this type of substances or this type of biological fertilizers, which is developed due to these scientific protocols, so they have an immense potential. It functions in long duration, production cost is very less, they are environmentally safe because they are not producing any kind of toxicity. Okay, and then residue problems. You know that chemicals are creating lots of residue problems, which is in case of suppose tea, it is more popularly known as maximum residue limits, MRLs. So as a result of which, you know, there are lots of tea while getting back from different kind of countries after even after export also. Okay, so these type of things actually happening due to lots of deleterious effects of chemical applications. So while with special reference to tea, if we see again, so soil compaction, this is one of the important issue in tea ecosystem, and this soil compaction is the result of chemical applications. And while application of this organic fortification or microbial fortification techniques with the use of these biological fertilizers, quality biological fertilizers, we can reduce the compaction, soil compaction. And whenever we are reducing the soil compaction, the soil becomes more fertile, the soil becomes, their water holding capacity becomes very high, and ultimately soil aeration takes place. Proper soil aeration takes place, and the plants become actually what happens? Plants comes back from stress. Okay, so adoption of quality biofertilizers and efficient microbial removal are certainly beneficial to the industry in reducing soil compaction. Another thing is that disease management. While for disease management, we have done some experiments. So this is one of the important disease problems, that is Pestilocopsis tea. This is responsible for causing grape blight in tea. So this is one of the important problems in foliage, tea foliage. You can see some concentric circles or rings in the leaves or foliages. Okay, so these foliages get hampered due to this pathogen means infection. And what we have done, we have isolated the CDMs, and you see B, left hand side B, the two photographs. This is the ultimate growth or a full plate growth of the Pestilocopsis tea. And whenever we are doing experiments in the D, figure D, so you can see after addition of our microorganism, Pestilocopsis tea, so it can grow well in our plates. So this is one of the most important aspect of disease management using the cellulose degrading microorganisms. So these are nothing but these are some of our large tanks prepared for field applications, and their growth and vigorosity has been noticed after 48 hours, 72 hours, and then 96 hours of their growth. And this is one of the important experiments, which is the degradation study. So rice straw has been accumulated, and then cellulose degrading microorganisms has been used here at different score concentrations, likewise 1%, 2%, 3%, 5%, and ultimately we are getting some very good results. So organic matter getting decomposed day by day. Okay, so this is one of the experiments where we are using cloning litters in our green pollinate bags, and it is deposited in the rhizosphere, in the tea rhizosphere, and after getting it, we are seeing their nutrient recovery content or compost recovery content. It is very high, it is good, and this is actually due to after preparation of the consortium, CDM consortium. Okay, this is another experiment where we are able to reduce the soil compactness in tea soil. As I have already mentioned that in tea ecosystem, soil compactness is one of the most important problems. And while doing this ring or core methodologies, we are adopting this cellulose degrading microorganisms under tea cultivation areas which is more soil compact, which are problems in soil compactness, and surprisingly our microorganisms also work well and cellulose degraders in affecting soil compactness, reducing the soil compactness. Okay, so integrated disease Integrated disease management. So this is another important asset while using the cellulose degraders. So IDM is basically designed to reduce the harmful effects of chemicals. Okay, so we have to reduce the load of chemicals also while in plant protection purposes. Plant protection is what? That is, we have to reduce the load of pests and pathogens in agriculture. So this is the overall integrated crop management schedule. So integrated crop management schedules, we have to care about energy consumption. We have to manage the shade tree and waste and pollution management. This is basically implied in case of suppose any kind of agriculture, but in shade tree management, suppose it is especially concerned with the tea ecosystem, then organizational management. We have to see all the things. No? So while adopting a strategy, we have to see all the things: cost effectiveness, environmental management, eco-friendliness. Okay, so we have to see all those things. So with the application of these organic management strategies, so integrated crop management is also growing day by day. Okay, so now escaping, our main aim is to escape the chemical trap. We have to now reduce the chemical trap, or we have to, means day by day we have to go from the chemical traps while reducing their load or while limitize their load, because until and unless we reduce their load, we can go beyond these agricultural practices. So there are lots of increasing costs. I have already discussed the different disadvantages while using the chemical pesticides or chemical fertilizers in any kind of agriculture. Okay, so likewise, so quality organic manure production or microorganisms. If we use the benefits of microorganisms in decomposition activities or degradation activities, the agricultural wastages, which is dumped day by day in each and every ecosystem area, so they can be used, they can be easily degraded for the production of some quality organic manures, and they can again be recycled in the agricultural systems. So this is one of the another experimental purposes or experiment which we have got in our mulching. You know mulching, I think. So this is normally usually used in tea cultivated areas where the young tea seedlings, after their growth up to 6 months, so the soil, you know that there are lots of temperature, no? So the moisture is actually retained, and moisture is usually retained due to application of this mulching material. And if mulching materials we have to treat with these cellulose degrading microorganisms, there is every possibility to load or to accept the moisture contents. Okay, as a result of which that we have seen that quality of production in tea is getting also improved day by day. So field application of microbial and enriched mulching materials prepared through CDM strains. And with this, I conclude my presentation for today. So thank you very much to the dear students for your nice listening, and hope you have also cleared some of your areas of specializations or some of your areas of expertise, why we should go for alternative technologies like non-chemical approaches in tea or non-chemical approaches in agriculture, especially for to adopt the sustainability in agricultural practices. So thank you very much.
Thank you, Professor. Students, the session is open for discussion. If you need any clarifications, you can ask Professor Pranav. Any questions? No questions? Okay.
I think students have, yes, this is actually very means basic level of presentation. I think students have got the points actually.
Yes. How
Yes,
bio-waste composting influence pesticide residue levels? One question is that.
Yes, actually what is happening, we all know that harmful effects of chemicals. Chemicals are related to their actually toxicity, and this toxicity results for some load, toxic chemicals load, no? Then what is happening while using this biological compost? So these are residue free. So whenever there is a residue free applications, so definitely there will be no problems of residue in ultimate, means in the finished product. Suppose in case of tea soil, when we are using organic amendments, or when we are using organic biofertilizers in the soil conditions, so ultimate in case of tea ecosystem, our finished product is what? Our black tea, green tea, or any kind of oolong tea, or any kind of finished product. No? So this finished product is not actually reported for the presence of any kind of residue. But what is happening in case of application of chemicals? As chemicals, we know they have some toxic chemical components. No? So they are getting residue problems. So this is why we should go for organic amendments or organic applications of these biological fortifications to enrich the product and without a residue problem.
One more question is there. In plant tissue culture, why most of the plants survive and grow on MS medium?
What, sir? What, sir? I am not listening.
In plant tissue culture, why most of the plants are surviving and grow on MS medium?
Humus media.
MS, Murasage medium.
Humus, HUMUS, humus.
No, Murasage, MS media, plant tissue culture medium.
Plant tissue culture medium. Actually, what is happening? Whatever media we are basically used in tissue culture, or whatever media we are basically using for growing the microbes, and then these media are actually, what is happening? These media are actually non-toxic. So that non-toxic medium, it eventually not incorporate any kind of residue problems. Okay. Suppose in case of tea also, we are also producing tea in MS medium also, and these MS mediums are free of any kind of harmful components. No? So likewise in our tea cultivation areas, in our means we are growing conditions also, we are using some natural media like potato extravaganza media. So these mediums are actually free, and whenever we are getting the plants growing, so the mediums are actually free of any kind of harmful toxic components. As a result of which we have no actually assumption, or we have no possibilities of coming some residue problems. Yes, any other questions?
Okay, you got your answers. Are you clear? One more question. If many side effects of chemical fertilizer, so why used in large scale?
There are lots of effects of chemical fertilization.
Yeah, side effects of chemical is there, fertilizer. Then why it is still used in large scale?
Yes, what is actually happening? Actually, now in case of, suppose in your tea ecosystem, if you assume as a role model, what is happening? So chemical fertilizers or synthetic fertilizers or organic fertilizers, these fertilizers are actually added to improve the quality of soil, because as plants will grow, normally the nutrients getting depleted day by day. And what is happening? These organic formulation strategies, they also need some time. Okay, this needs some time, then some protocol standardization, and then thing is that sustainability improvement. This is also another important protocol, and which need to be for the long time of for long proportions. Okay, and then chemical fertilizers, chemical formula is there, so you can procure them. Okay, or you can manufacture them easily actually. And but what is happening now? Enter world, and our Prime Minister, Mr. Modi, is also now in favor of the use of some sustainable resources or sustainable management practices, using the use of microbial fortifications, using the load of botanicals. Okay, so we have to reduce the load of chemicals, although it is used long back or still it is in use, but we have to reduce the load of chemicals with the use of this type of our quality compost or quality biofertilization production. Okay, until and unless if we don't use it, our agricultural system will be ruined, and our soil fertility will be ruined. There will be adverse or deleterious effects. So for this reason, we have to go day by day, and we have to adopt the organic management practices actually. Although the chemicals are also there in the market.
Any other questions? One more question.
Yes.
Is there a database available for determining the toxicity of chemicals?
Is there databases for data?
Determining the toxicity level of chemical.
Yes, why not? In each and every agricultural crop, there are some data monitoring system, and it is known as your actually it is your CIB registration, Central Insecticide Board registration. So suppose in case of tea, what kind of chemicals can be used, or what kind of chemical can be used in what dose, or what is the recommendations? So all the data are actually available. So likewise, in case of tea ecosystem or in case of tea applications, we have our tea board. Okay, so we have our tea board. So it is known as your plant protection code. Likewise, in case of coffee also, they have their own code of application of chemicals. And you can use beyond the load of, or beyond the recommended dose of chemicals. And the chemicals, whatever be the insecticides, whatever be the fungicides, whatever be the acaricides, what may be the use of the chemicals, they are already listed in their what code, that is the plant protection code. And for each and every agricultural crops, it is in our legislation, or it is in our under different board management systems, or in our agricultural crop system. So there is no worry fact actually. So we can see what are the chemicals, what are chemicals are available for application, what are their standard dosage. Okay, so these are all actually available.
Any other questions?
Excuse me, sir.
Yes, go on.
Yes, sir, I have a question. So how about the use of hydrogels in the agriculture? Can it solve the most of the problems that our agriculture is facing today?
I'm not actually clear about the question.
Can you little bit louder?
Yes.
Use of hydrogels in the agriculture.
Use of hydrogen in agriculture, yes.
It's hydrogels, sir, hydrogels.
Acha, hydrogel, yes.
So how about using the hydrogels in agriculture, sir? Can it solve the most of the problems?
Actually, what is happening nowadays is there is another technology of application of microbial fortification in hydrogels. Okay, so hydrogels, for improving the quality of hydrogels, for improving the quality of hydrogels in agricultural system, there is now technology has been developed in ICR, Indian Council of Agricultural Research also, IARI also. So there are lots of technologies for improvement of this hydrogen. Okay, so due to these microbial fortifications, and these are usually added for integrated nutrient management practices. Okay, so there are lots of microorganisms like Bacillus, Pseudomonas. Okay, so then these microorganisms can be used for the preparation of microbial fortified hydrogens, which can improve the quality of soil. And if the soil quality is improved, obviously the plant health perspective is also getting improved day by day. So there is a technology, and it's developing. These skills are actually developing. It's very good questions from you.
Thank you, sir.
Yes.
Any other questions? If no more questions, then we thank Professor Pranab Bhattacharya for joining us today and elaborating the sustainable agricultures and microbial, how microbes can help us in decomposition and transition of soil ecosystem. Thank you so much, Professor, for joining us today.
Thank you, Dr. Murugan. It's your effort while conducting such type of these programs for the improvement of the quality of our students at the national levels. So I am also thankful to you and my dear students. Go ahead and blessed wishes for you in the coming days. Yes, thank you very much.
Students, okay, that's all for the day. We will resume back tomorrow with another set of lectures. Thank you for joining us.